good morning, good afternoon, good night, whatever time zone you happen to be working from. For me, it's a nice early morning right now. Welcome to today's webinar on an introduction to flow cytometry. This webinar builds to some degree on the webinar that I gave last week on an introduction to fluorescence in general as a measurement and debugging tool and to plate readers. I'll be recapping some of the material from last week briefly for those who may not have been here, but this should be able to mostly stand on its own. For things where it might be useful to in-depth see material from last week, I'll point that out when we get there in case you haven't seen it. But right now, let's get going with starting with an introduction to the concepts of flow cytometry as we begin this summer webinar on quantifying fluorescence and cell phenotypes with flow cytometry. So, calling back to one of the things we talked about last week in the general introduction to fluorescence, there's a range of instruments that people typically work with. Fluorimeters or plate readers only give you population level measurements, but plate readers can give you very high throughput time series. Fluorescence microscopes can give you beautiful resolution, but only a relatively small number of cells, not a huge amount of dynamic range in looking at the actual intensities. Today, though, we're going to focus on flow cytometry, which is my personal favorite instrument. This is an instrument that gives us potentially very high throughput. A lot of flow cytometers have plate modes where they will automatically run through a 96 well plate and give you measurements for every well on the plate in fast order. They give you single cell measurements, which are extremely valuable, as we'll talk about in a minute. And a huge number of single cells. You can easily get 100,000 or more single cell measurements in just a couple of minutes from a sample. So you can get a fantastic amount of information about the individual cells in your samples. And these instruments have a great dynamic range. Depending on the instrument and the particulars of the cell and fluorescence you're working with, you can get 3 to 6 orders of magnitude difference between the highest and lowest values that you can quantify. The only real problem with a flow cytometer is that it's a disruptive measurement, and therefore it's generally not as good for doing time series measurements as the less invasive ones. I'm not going to talk much in general about fluorescence this week, since I covered that last week. If you want to know more about the background of how fluorescence works, what it is, I recommend that you go take a look at the recording from last week's seminar, which will hopefully be up if it's not already, and the slides are already posted. Well, let's dig into flow cytometry and why flow cytometry first. And really, it comes down to the fact that the distributions of your cell behaviors matter. Right, let's take a look for a very simple example at these two groups of cells here on the left and on the right. These two groups of cells have the same mean fluorescence, but they're clearly not the same. The ones on the left are very uniform. The ones on the right are extremely bimodal. If we want to understand what's going on in the cells that we're engineering, we want to know whether we're looking at cells like the cells on the left or cells like the cells on the right. So really, single cell measurements matter because they tell us not just what the mean cell is, but what the distribution of different behaviors. And with a lot of single cell information, we can get into pulling out a huge amount about the different parameters of a model and fitting those to try to understand very deeply what's going on. I've got a couple of examples here of particular places where I've used modeling based on large numbers of single cell measurements. On the left, we were doing some work with RNA replicons that you transfect into cells and then they replicate within the cells. You may have heard about some of these recently. These RNA replicon approaches are one of the key technologies that's being used in some of the COVID vaccines that are in phase 3 trials right now, where you put stuff into a cell and it amplifies itself up because you've used part of the virus, but not all. And to understand what was going on here, we needed to understand a bunch of things about how efficient the doses were at getting things into cells and how much variation there was from cell to cell and how these expressed, how the doses got, what the actual initial levels were that we could see as a quantization effect, how things varied from cell to cell when we were dosing in 2 different replicons at once. And all of these were parameters that, although we wouldn't have been able to see them, we were just looking at the mean. When we were looking at a distribution, we could pull out a model and say, if it's low initial doses, we should see more spread down on this axis. When we're looking at the things that change because of the cell rather than because of the replicon, we should see it stretching out on this diagonal axis. The fraction of cells that don't get anything in them at all on that bottom left tell us something about the efficiency of the dosing regime. Another example that we'll actually see more about later on the right is understanding things about the operation of our cells and our instruments based on a simple constitutive fluorescent protein going in. This is going into some HEC293 mammalian cells. And here we're, again, pulling out a whole bunch of parameters. We're pulling out the fluorescence overlaps through this spectral bleed measurement over on the right, this diagonal line coming up. We're being able to, from this bimodal left versus right red blobs, we're being able to figure out what the efficiency of the transfections is and what the level of variation between cells is from the right-hand blob, and being able to figure out things about autofluorescence from the upward distribution. So again, a huge amount of information potentially possible, but to get this and to be able to fit these models and have confidence, we need lots and lots of individual cell measurements, which is what a flow cytometer can give us. One more motivational note is that being able to look at the distributions can also really help with quality control and making sure that your experiments are working the way that you want them to. Here's a couple more examples of looking at the structure of the distribution on the left. Even though the cells are covering the same range and the actual mean fluorescence isn't very different between the bad samples and the good samples, those bad samples sort of trailing down at the top distribution told us that something went wrong with those samples. We didn't necessarily know what. Similarly, we could look at a distribution of the structure of the high versus low on the right and say this whole batch went wrong. So another motivation for being able to get single cell data. So how does this actually work? Brief reminder about how fluorescence works again. If you were here last week, you will have seen this before, but fluorescence basically works by, we have some molecule, here's a GFP in the middle here, which incoming light excites that molecule and causes it to resonate across a wide range of frequencies as it emits light back out again. Typically, the excitation is shorter wavelength and more energetic than the emission. In a flow cytometer, at least, typically that excitation is monochromatic, meaning it's coming from a laser and it's very precise. Fluorescence, reminder, gives us a great signal that's highly proportional to the number of molecules, but the exact relationship between the strength of emission that you're getting in context is pretty sensitive, hard to calculate. There's a whole bunch of different factors that can come in there, so we always have to calibrate it. Later today, we'll talk about how to calibrate for a flow cytometer, where there are some fantastic tools to help us. And when we use more than one fluorescent protein, we want to make sure that they have good spectral separation so that those emission blobs are not overlapping each other a bunch with the excitations that are being used for each. So how a flow cytometer actually works is that our cells basically get sucked into a microfluidic system where the sample gets sputtered one by one out a nozzle through a path of one or more lasers. In most modern flow cytometers, you have multiple lasers, which is great because each laser is a different emission. The cell passes through the laser, and as it passes through the laser, light gets scattered off of the cell at a bunch of different frequencies. And that ends up giving us 3 sets of measurements. This forward scattering measurement gives us particle size. The side scattering measurement gives us a measure of something that it doesn't translate terribly well to physical properties. It's often referred to as sort of granularity. It's something about how lumpy the cell is visually. It's not used for terribly much besides figuring out what is a cell and what's not. And we get a bunch of fluorescence measurements. In addition to the general problems that you have with fluorescence, there are some instrument-specific challenges. The first of which is just that you're dealing with a huge number of data points. So you really have to bring in some good analysis software of some sort in order to handle 100,000 data points per per sample. You can take less, but if you take less than 10,000, you're really not taking advantage of your flow cytometer. And the more data points you have, the finer features of the distribution you'll be able to see, and the more information you can pull out for your models. Another challenge here is that, again, this is both a benefit and a challenge, that the individual cells you're looking at will behave often very, very differently from one another. The whole point here is we get to see that. But it means that you're going to have to think not just about a mean, but about understanding the structure of the distribution. As I noted, it is a disruptive measurement because these cells are not sitting nicely in the colony where they were before. Especially if you've got adherent cells, you got to break them off the colony floor or walls, and the cells will get unhappy about that. So you basically have to throw away anything that went through the flow cytometer. And not everything that goes through is a cell or is the cell that you want. You also will get measurements from debris that you need to filter out, from clumps, and you can even get, if you have pipetting issues, you can get contamination from one sample to the next sample. We'll see how to handle some of these things as we go forward. But despite these challenges, a lot of these challenges are about the sheer fact that this is an incredibly powerful instrument. And like I said, this is my favorite tool. It gives you so much data and can link you directly to modeling in a way that something like a plate reader just can't. Here's what a flow cytometer looks like under the hood. One particular model, there's a lot of models that look a lot of different ways. This one happens to be in the Soro lab at the University of Washington. Over here on the bottom right, this little black tube going into a sort of shiny metal connector that's where the laser is actually coming in on this machine. And then these little colorful blocks are mirrors, and they're mirrors that also act as filters. So we've got first a notch filter that's taking out the laser. This is a 561 laser. That means it's essentially a reddish laser. And then the beam is getting split to send the high frequencies up and the low frequencies forward. Those high frequencies are going to get sent through a band pass filter and another to in another path to 2 detectors here. Over on the right, we've got a mirror that's just splitting the beam into 2 different detectors. And this detector over here, number 5, doesn't appear to be getting used in the current configuration. So you can see this is a very modular machine. It's even got a whole bunch of spare splitters and filters up in the slots at the top. So you can configure these around for all sorts of different, very specific measurement patterns if you want in a lot of cases. Not all machines are quite as friendly as this one for reconfiguration, but a lot are. So moving forward, let's talk about one of those challenges with a flow cytometer, that a lot of the data points that you get are not from cells. Here's one example of a fairly typical distribution. Here we're plotting the forward scatter. This is the thing that has something to do with particle size, against the side scatter, which is the thing that has something to do with particle sort of lumpiness, granularity. And what you'll typically see is that you've got one sort of nice, tight, high density distribution. Here, the hotter the color, the more data points are in that place. And typically your nice, tight area is where your cells are. And then you've got other stuff that's bigger and more complicated, which is likely to be clumps of cells. And you've got other stuff that's smaller or just scattered all around, and that's likely to be debris. That's like fragments of dead cells, or bits of precipitated media, or just junk that's floating around in your flow cytometry machine from the person who used it before you and didn't clean it properly, or who knows what. So one of the things that we almost always end up doing with a flow cytometer is what's referred to as gatoring, which means filtering out all the stuff that we think might not be cells. We'll talk more about how to do that in a little bit. Let's talk about one more thing about how the instrument works. When those cells are actually passing through the laser, that's not an instantaneous thing. It happens pretty fast, but what actually gets measured is a pulse, where as the leading edge of the cell or other particle goes into the laser, then the fluorescence response goes up, it stays up, and as it comes out of the laser, it goes down again. And a lot of instruments, most modern ones in fact, will report 3 properties off of that pulse. They'll report an area property, which is what's the total amount of energy that came out during that pulse. They'll report a width, which is how long did it take for the cell to go through the laser. And they'll report a height, which is what's the maximum intensity that happened at any point. And these usually get abbreviated as -A, -W, and -H. So if I'm measuring on the fluorescein channel, which is often labeled as FITSI, I might have FITSI A for area, FITSI W for width, and FITSI H for height. Area is the one that we are typically most interested in, because that's the one that actually tells us how much total fluorescence there was in that particle. Width and height depend very strongly on how the cell is actually flowing through, so how fast it's going. There's different settings you can have in a machine about speed, about the orientation of the cell, things like that. So they're mostly just useful for gating out doublets, debris, and clumps. So for example, you can often detect a doublet, i.e. a pair of cells stuck together. If they happen to go through in the right orientation, you can detect it because you get twice the area and twice the width, but only the same height, because you've got sort of a stuck pair. But if they go through in a different orientation, you won't see that. So it's only so good. So bottom line, think mostly about area, but width and height can be useful for gating. One more key thing about gating. There's a little bit of a challenge in figuring out the degree to which you want to allow the instrument to do gating for you. Most instruments have an internal gating setup of some sort that allows them to record only some events and not others. So you can set up your whole gating system inside the instrument and have it only record the things that a priori you think are cells. That is typically a very bad idea because you're running the instrument without necessarily having calibrated it yet. And when the instrument is just throwing away data, if you made a mistake in how you set your gate, those events are gone. You will just lose data. So it's usually better to gather, to set your filters conservatively so that you end up with extra events that you can filter out via software rather than just not having the data in the first place. However, if you let your instrument try and record too much, and you let that its trigger for when it wants to count a pulse be too sensitive, then you can actually have too many pulses coming too fast and end up overwhelming the speed at which the instrument is capable of recording and losing a bunch of cell events. So what you end up typically wanting to do is to have a trigger set on your forward scatter for particle size, because you want to basically throw away the really tiny ones. Set it, based on you take one run of some simple junk, set it very, set it to some level, look and see if it looks like you've got cells getting cut off or if you've got too much junk, and set it so that your forward scatter is going to take only particles that look like they have a decent chance that they might possibly be a cell, so that you're not going to drop cell events, but you're not going to overwhelm recording capability. This is usually not too hard. There's usually a pretty clear gap. I see a couple Q&As. Question about, can a pulse be compared or set to a certain frequency? So this is a different type of pulse than that, where we're talking about just a signal that goes up and down as something goes through a laser. So it's not really a frequency here, it's just a single blob. And see, are there any especially interesting ways that machine learning or deep learning can be integrated with flow cytometry? I really can only just say yes. There's so much data, and people really haven't even explored that fully yet. I just recently saw a preprint from some colleagues of mine at NIST who are doing exactly this. And so this is something that I would encourage you to go and play with. But before you go and get into that level, we're going to want to make sure that the data that goes in is clean, which our next segment is going to be about. All right, one last sort of instrument level thing for making sure that our flow cytometer is going to do what we want is it's important to be aware of the size of the cells you're working with versus the size of cells that your flow cytometer was designed for. Flow cytometry really originally came out of the medical community, mostly designed for human cells. So flow cytometry is very often used, for example, for looking at separating out, say, red blood cells and white blood cells and different types of white blood cells to give blood counts, things like that. Those are pretty big cells, like 10 microns wide. So if you're using really small cells like bacteria, as many iGEM teams are, some instruments will run into problems where they're just not quite set up to wash cells that small out well. And here's an example down at the bottom of a case that we encountered a few years back with this, where you can see, here's well A6 with this blob over here. Well A7, this is clearly the stuff that's supposed to be in well A7. And there's a little blob of well A6 that made it in, about 1%. Well A8, is supposed to be a replicate of A7. It's looking pretty much the same. There's a tiny little fragment of the A6 left. So if you run into something like this, you can play with the settings to try and improve your wash, and you can even add wash wells between your samples. That's what we did to correct this problem here. On the opposite side, if you start running enormous cells like muscle cells or neurons through your flow cytometer, you will clog the flow cytometer nozzle, and you will make the people who own the flow cytometer very angry at you. So don't do that. When we've run into that in the past, one of the things you can do is switch from using fluorescent proteins that are dispersed through the whole cell to fluorescent proteins that localize in the nucleus and use a nucleus extraction protocol. One last thing, some groups have strict rules about what cells are allowed to go into their flow cytometers. Like you may not be allowed to put fungi onto a flow cytometer that's reserved for bacteria or vice versa. So remember to ask the people that you're working with, whose flow cytometer you're borrowing, if your cells are allowed on the machine at all. Again, that's not a technical point. That's a relationship note that's very important. So in summary, for this first chunk, flow cytometry gives us lots of single cell measurements. It's great for that because those distributions of single cell behavior reveal all sorts of things about parameters of models that we might want to make, about how our system works that allow us to get deep understanding and really get in and debug and control our systems. They also reveal all sorts of protocol problems. Most flow cytometers measure multiple fluorescence channels, as well as the forward and side scatter channels on 3 properties: area, width, and height. Forward scatter tells us something about how big the particle is that we're looking at, which is useful for understanding if we're looking at a cell or not. Fluorescence is generally best measured with area, not width or height, but width and height are often useful for gating, especially width and height on forward and side scatter. And if you're working with very small cells like bacteria, on an instrument that was not designed for bacteria, beware of well-to-well contamination. So let me pause here and take a look at the Q&A. Are there any free visualization softwares doing the job of FlowJo? Yes, and we are going to talk about some of them in the next section, so that's a great lead-in. Is there any predefined angle between the laser light and the detector for fluorescence quantification? Not quite sure what you're going after there, but to the best of my understanding, the actual structure of the light path is very instrument dependent. And because of the nature of how lasers work, essentially, you're basically sending stuff on a straight path and just bouncing it off of mirrors. So in theory, you should always be hitting it straight on. So like in that look at the machine we saw under the hood, that was all straight lines and 45-degree angles. So it should be hitting with no angle between detector and laser. And that's what you want, because that's going to maximize the signal that comes in.